Hello, everyone. Welcome to another session of the Porous Media Tea Time Talk. Today it's a very special one for all of us. It's a session number 50, and I think the whole team is really surprised that this was originally intended to be kind of like a short project to help people present during the time of the pandemic when like many conferences were being shut down. But suddenly here we are 3 years later and 50 sessions later, and I think about maybe almost 100 speakers later. So it's been quite a pleasure, and I think we're very happy to see that the community still welcomes this initiative so very much. So my name is Marcel Morer. I work at Por Lab at the University of Oslo, and I am joined here by my friends Mohammed Nurepu, Mahdi Mansouri, and Federico Lanza. And together with the whole team behind the Porous Media Tea Time Talk, we would like to welcome you to this session. I want to share a little bit here before we start. So this is, today I've put like here the whole team, the people that are currently on the team and also the ones that came before that started with us, to thank all of them for their contribution, for their enthusiasm, their input into this project, in particular to Maya here. She started the whole thing, but she triggered the idea and convinced us to join for that. So that's very nice. And I also want to thank Interpor for helping us along the way, for always being a friends of a Tea Time Talk. We have been using Interpor to spread the message since the very beginning. They have always been very supportive of it. And now together with the Young Academy, this has been helping a lot the projects to grow and spread. So enough of welcoming you all. We're going to have a bit of a different session today. So normally we have only PhDs or postdocs or early career researchers for the Tea Time Talk. This is kind of like the idea of the project. But today, since it's number 50, we kind of decided to turn the rules a little bit. And we invited two researchers that they are not like super early careers as the other speakers were, but they are also not like a very senior. So they are kind of like they have made this transition from early career to staff to permanent positions not too long ago. And we invited then Dr. Hang Dam and Dr. Amir Pahlavan to talk about not only their results, but also today to focus a little bit on their career path. So we will welcome everyone to join in on the discussion. We'll do things a little bit differently today. We're going to have the questions that you type in the comments field on YouTube, so you can type it in at any time. But we're going to read that at the end of both presentations, because then we have kind of like a question session together for both of them, both about the research that they're going to present, but also about the career path. That is kind of like the goal for today. Okay, so starting with Dr. Hang Dang. So Hang, if you can put you in the stage, and if you can like have your presentation ready there. So Dr. Hang Dang, she's an assistant professor at the College of Engineering and an adjunct professor at the Institute of Energy and Institute of Carbon Neutrality at Peking University. She received her bachelor's degree in Science and the Arts from Peking University and her PhD degree from the Department of Civil and Environmental Engineering at Princeton University in 2015. She worked in Lawrence Berkeley National Laboratory's Earth and Environmental Sciences area, first as a postdoc fellow and then as an earth research scientist. Her recent research centers around reactive transport processes of various fluids and chemicals in fractured and porous media and pore-scale interactions between multi-phase flow and geochemical reactions. She currently also serves on the editorial board of the Journal of Contaminant Hydrology and the inaugural Early Career Advisory Board of Environmental Science and Technology. Today, she'll talk about modeling reactive transport processes in porous media. And Han, thanks so much for being with us here. The floor is yours.
Thank you so much. It's such a great pleasure to be part of the 50th Porous Media Tea Time Talk. And as mentioned by Morel, that I will have two parts in my presentation. First, I will go over two examples of my work on modeling reactive transport processes in porous media, and I will share a little bit about my experiences as I develop my career. So so this community is all about porous media. It's everywhere in our life. My focus is a bit about porous media in Earth systems that are important in providing energy and resources to human society. And in these systems, the structure and chemical physical properties of porous media actually evolve as a result of geochemical reactions and the coupled hydrological, thermal, and mechanical processes. And here I'm showing two examples. One is on the left is the geologic carbon storage, where we have CO2, water, and sometimes contaminant gases interacting with reservoir and cap rocks. And on the right, we have the example of unconventional shale gas development, where we potentially could have scaling in the fracture and in the borehole. So it is important to understand the interactions of geochemical reactions and other processes. And the reactive transport model is one of the most powerful tool that help us investigate the intimate coupling of flow, transport, and reactions in porous media. And the figure on the right basically is a summary of the publications over the last 3 decades that used reactive transport model as the method. And also you can see how this tool has been used to investigate issues across many different disciplines, including energy fuels and environmental engineering, etc. So there are 3 different, typically, we talk about 3 different types of reactive transport model. We have continuum scale model, where we don't explicitly track the geometry of the porous media, and we use averaged properties such as porosity, diffusivity, and surface area, effective surface area, to model the system or the evolution of the porous media. On the other end, we have pore scale model, where we explicitly track the geometry of the porous media. We solve for the velocity field, and we also treat mineral reactions as a boundary condition. Those are some key features of the pore scale model. And we also have a hybrid model, and here the equations are the Darcy-Brinkman-based hybrid model. This type of model sort of combines some of the advantages of pore scale and continuum scale models. In this case, we can represent the geometry of the porous media using a microporosity field, where we have 1 for the pore space, 0 for the solid, and a fraction value for the interface. So it's easy to track the geometry as well. And one of the overarching objectives of my research is to develop predictive understanding of the alterations of porous media driven by geochemical reactions. And today I will be giving 2 examples. One is about precipitation-driven alteration of porous media, and the second is about understanding reactive transport processes in porous media, where we have 2-phase flow. So for the first example, precipitation, we care about precipitation not only because it is an important process, as mentioned before, in shale gas systems and many other systems, but also because it is a very interesting problem to work on. Mineral precipitation, we can't really simply treat it as the reverse process of dissolution, because it involves nucleation, crystal growth, and etc. And they have a very complex dependence on fluid chemistry, the substrate properties, in addition to flow and transport. And this figure here is an example showing the morphological differences when the substrate is different for the precipitation. And in order to account for the impacts of fluid chemistry and substrate, we typically use the classical nucleation theory that can be described by the equations shown on the right. So here for the precipitation-driven alteration in porous media, I will also be talking about 2 examples. One focuses on precipitation and the effective diffusivity of the porous media. This is really motivated by some previous experimental work by our collaborators in France. So this is a column experiment where we have strontium and sulfate-rich solutions diffusing into the column, and a narrow precipitation zone formed, as you can tell on the city image highlighted by white. This precipitation only changed or reduced the porosity of the column by a very small magnitude, but the drop in the effective diffusivity in the column is huge. And this drop was not able to be captured by the continuum scale models. So we took a pore scale try of modeling this same experiment. And what you are seeing here on the left, the video is showing homogeneous nucleation. So basically the precipitates form in the solution. This is also a mechanism that has been reported in previous microfluidic experiment. And on the right, this is an example of surface growth where we see precipitation occurring and coating the mineral grains or the primary mineral grains. And this is another mechanism that has been reported in experimental observations as well. So basically here, by accounting for different mechanisms of precipitation, we are able to reproduce different alteration patterns observed experimentally. And taking these two different approaches, and we modeled different initial geometries, different types of geometries, and considering uncertainties in precipitation rate. But basically what we observe here is that regardless of other differences, there's always a critical porosity value that the system reaches. Afterwards, the system stops evolving, or the porosity stops decreasing. And this type of sharp decrease this type of lack of change in porosity is also correlated with a sharp drop in the effective diffusivity in the system. And this type of sharp drop in effective diffusivity was not captured by the traditional Arches law as shown in red. And based on our pore scale simulations, we were able to revise this relationship to account for the precipitation dynamics. And this updated relationship was able to provide us with much better match with the experimental data. So basically, we took this relationship into our continuum scale model, and now, voila, we can reproduce better the experimental observations. And the second example is about cold dissolution and precipitation. And this type of system, basically the dissolution of the primary phase, which is celestite here, provides the necessary chemical, which is sulfate here, to precipitate the secondary mineral phase, and it's barite in this system. And again, this is some experimental observations from my collaborator, Jana Punusamy. It's also a column experiment, and what you're seeing are SEM images taken at different spots along the flow direction for different oversaturation states. So basically what they observed is that for very high oversaturation, the precipitation is quite uniform, whereas with the intermediate oversaturation, the most significant precipitation occurs in the middle section. And for the lowest oversaturation, the precipitation or the replacement of the primary phase by the secondary phase is very highly localized at the inlet. And they also tried to model the experiments using continuum scale models. And again, unfortunately, they were not able to reproduce the patterns and the fluid chemistry. And no surprise, we were able to better reproduce the experimentally observed fluid chemistry and precipitation patterns once we take a pore scale perspective or take the pore scale approach in the modeling. And since we don't have the actual geometry in the column, we assumed that we are following basically a streamline, and then observe the precipitation patterns along the streamline or along the flow direction. And what you can see is much better match in terms of the fluid chemistry. And we also captured the overall pattern where we see relatively uniform precipitation, high or larger precipitation in the middle section, and also localized precipitation at the inlet. And we also kind of modeled the grains, a spherical grain at the inlet for different oversaturation states. And again, what we're seeing is comparable coating of barite for the two higher oversaturation states as also observed experimentally and reproduced in the streamline simulations, and much thicker coating of barite in the lowest oversaturation state. So again, this is consistent with the experimental observations. And another feature of precipitation is the probabilistic nature of the nucleation process. And here we took this into consideration and tested the impacts of different fluid chemistry over saturation state, and also nucleation density of the substrate. So that's determined by the property of the substrate. And we could see significant difference among different fluid chemistry and substrate properties in terms of the microporosity in the precipitates, and also the surface coverage on the primary phase. So just a quick summary. These exercises really confirmed that in order to reproduce or capture the pore structure alterations observed experimentally, we need to account for the interactions between flow, transport, and reactions at the pore scale. And also using the classical nucleation theory and the pore scale model, we were able to show that the texture of the precipitates is sensitive to the transport properties, the supersaturation, and also other kinetic parameters that are related to the substrate. So moving towards the second example about multi-phase flow, and this is, again, quite common in a lot of the Earth systems, such as the geologic carbon storage, the geothermal, and the Earth's critical zone. The interactions of multi-phase with geochemical reactions would shape the evolution of porous media, but in most of the continuum scale models, we assume that multi-phase flow doesn't really impact mineral reaction rate, or the impact is simply accounted for by a power law function of water saturation. And here you can see one example. But whether this type of relationship, which is highly empirical, is valid, is something that we are not certain. And conceptually, we know that the two-phase flow or multi-phase flow would definitely impact where the reaction occurs and how fast the reaction occurs. And what the example is going to show you is one way of accounting for or quantifying the impacts of two-phase flow on reaction rate, and also whether there's a relationship to be found between this reaction rate change and water saturation. So for this purpose, we developed a pore scale modeling framework coupling the OpenFOAM library and the geochemical code CrunchTope, so that we can simultaneously account for two-phase dynamics and complex geochemical reactions. And basically in this framework, the OpenFOAM is driving the time stepping and solving for flow and transport. And in each time step and in each grid cell, we would call the geochemical engine, which is Crunch here, to solve for the aqueous reactions and the mineral reactions. And specifically in this example, since I will be talking about two-phase flow, we're using the built-in solver Interform. For the geometries we considered, it's just a very simple 2D pore doublet, kind of the simplest. It's inspired by the microfluidic experiment by Peking, we made some simplifications. And basically, we have two channels in the system. The top one is either the same or narrower than the bottom channel. And in total, we have 15 geometries. We also considered a combination of wetting and non-wetting velocities, giving us Reynolds number and Kepler number are ranging across 1 to 2 orders of magnitude. What is highlighted in the red box is the reactive zone. And basically here we are considering a quite simple reaction, calcite dissolution in water that's saturated with CO2 at the atmospheric conditions. In addition to the two-phase simulations, we also simulated the corresponding single-phase case where we have the same total flux so that we can compare to what extent the reaction rate has been changed by the two-phase dynamics. So what you're looking at here is the time series of 5 different geometries where we see the reaction rate for both single-phase and two-phase cases. So the co-injection of water and gas would lead to a series of gas bubbles going through the pore doublet. And following the steady state of the hydrodynamics, we also see the steady state of reaction rate. But for the two-phase case, the reaction rate is much reduced compared to the single phase. And this is partly attributed to the reduced wetted surface area because as gas bubble migrates through the system, that surface is no longer in contact with the reacting fluid. But quantitatively, the reduction in wetted surface area is not able to account for all the changes in reaction rate. So the other contributor is actually transport limitation. So basically, we see in two-phase systems a smaller thermodynamic driving force as a result. And since we are interested in comparing the reaction rate change with water saturation, here we pulled together all the data points, plotting on the x-axis water saturation in the pore doublet and on the y-axis the ratio between the reaction rate in the two-phase case and the reaction rate in the single-phase case. So we see some sort of trend in the data, but it's quite scattered as well. And if we follow a given symbol, which would be the same velocity but different geometries, or if we follow the same color of symbols, which would be the same geometry but different velocities, we see non-monotonic relationship. So this means this is really cautioning us in developing a relationship between water saturation and the reaction rate change. So probably the traditionally used power law relationship is not really the ideal or the optimal constitutive relation to be used. And another very interesting observation from these pore scale simulations is how the two channel, the reaction rate in the two channels vary in the single phase and the two phase case. So basically in the single phase case, we see the ratio between the reaction rate of the narrow channel and the reaction rate of the wide channel being always smaller than 1. So this means that over time, the difference between the narrow channel and the wide channel is expected to grow. So there's some sort of self-enhancing. Whereas in the two-phase flow case, we see the ratio being larger than 1 in most of the cases, meaning that over time, the difference between the two channels or the geometric difference in the two channels are expected to decrease instead. So there's some sort of homogenizing effect occurring in the two-phase system. So quickly summarizing, we see that in two-phase cases, the reaction rate is actually reduced, partly because of the reduced wetted surface area and partly because of the transport limitation. And the change in the reaction rate does not necessarily follow a power law relationship with respect to water saturation, because there's some sort of non-monotonicity in the relationship. And also we see how the two-phase flow dynamics is actually affecting the evolution of the pore doublet, which has also been observed in previous experimental studies. So that's the first part of my presentation, more about some recent work that I've been doing. And now I'm moving to the second part where I wanted to share a little bit about my career path. And when we think about career development, we typically think about us climbing the career ladder, which is sort of depicted on the sketch here. But I think that's one way to think about climbing the career ladder, but there are different ways of thinking about it. To me, there are two important differences for climbing the career ladder. The first one is that there's more than one ladder for our career development. There's research, there's also service and other stuff. And the second part is that we are not alone in climbing the career ladder. There's a community. So, okay, so let's move into a little bit detail. As mentioned earlier, I started at Peking University and now I've returned to Peking University. But in between, there are two stages of my study and research that are really important, which I think have really shaped who I am. And I will be talking a little bit about those two experiences. One is about my graduate study at Princeton University. I joined Princeton University, or I started my graduate study at Princeton University in 2009 with Professor Catherine Peters. And my work was about geochemically driven alteration in fractures in the context of geologic carbon storage. And there are a few important events or I would say important kind of milestones that occurred during my study at Princeton University on two tracks. The one is the research track. There are three important things. One is about the internship that I did at the National Energy Technology Laboratory. So that experience offered me the opportunity to get very hands-on with their facilities, like the big core flooding system and their industrial micro-CT scanner. So that's more than 10 years ago, and I was able to run in situ flow-through experiments using their facility. And that generated a lot of useful insights and data. And the second important thing that occurred to me is the Science, Technology, and Environmental Policy Certificate. So that's a program that offers graduate students from engineering and sciences to develop a deeper understanding of scientific, technological, and environmental and policy landscapes. And that allowed me to take my understanding of the physical systems of geologic carbon storage into understanding how, if we have any leakage from geologic carbon storage, that's going to impact the application of CCUS in our global energy system. So that's really a fun experience. And the third important, I would say, experience was about getting the opportunity to work at Brookhaven National Laboratory. So that's when and where I was able to combine microfluidic experiment with synchrotron-based techniques. So that's a really eye-opening experience. And in addition to that, I was able to get engaged with different student groups. I was part of the Princeton Energy and Climate Scholars. It's a student-run group. Every year we have the opportunity to develop a project, and this photo is showing us traveling in India, trying to understand their environmental and energy landscapes. And the other example is China Energy Group, where we kind of more focused on the issues associated with China. And after my graduation, this photo is actually taken right after my defense. I got a postdoc position at Lawrence Berkeley National Laboratory. I have two postdoc supervisors, Dr. Carl Stiefel and Professor Donna DiPaolo. And when I was at Berkeley Lab, the first important experience is being part of the Center for Nanoscale Controls on Geologic CO2. DOE funds several energy frontier research centers, and this is one of them. And this is the first time that I was exposed to team science. I was able to kind of start to understand how team science works and how collaborations across universities and across different disciplines in national laboratories can be forged. And the second important step while I was at Berkeley was being part of the early career LDRDO research program. And this, in this program, they took the cohort, they really kind of go out of the way to help us understand how national laboratory works. So that was very valuable experiences. Actually, when I first started at Berkeley Lab as a postdoc, I felt a little bit lonely because unlike my graduate study where we had tons of student groups, there was not really a tight postdoc community. So with several other postdocs, we started this Berkeley Lab Postdoc Association, and it was very helpful in terms of kind of social networking, as well as kind of get to know postdocs working in different disciplines. And later, I also joined the Women Scientists and Engineers Council, and that offered me the opportunity to work on different aspects of diversity, equity, and inclusion. And while I was at Berkeley Lab, I had so many different mentors. Some of them probably don't even know that they are my mentor. And one of them is Dr. Susan Hubbard, who's a great role model, a great role model for female scientists. So later, I also got engaged with other professional services, serving on advisory board, editorial board, and conference experiences really helped me understand how journal publishing and also professional societies work, because those are very important components of our research life. And I also got the opportunity to get engaged in scientific writing beyond research publications. So these are some examples of viewpoint articles where I had the opportunity to reflect on topics that are related to research, but not necessarily on a specific research topic. And also last year, there was a special issue on with Journal of Hydrology promoting women mentors in hydrology. And I worked with several other academic sisters writing a paper for or about our PhD advisor, Professor Catherine Peters, because she's also a very effective and a strong mentor role for all of us. So just a few reflections about research. I mean, these are kind of my reflections, not necessarily working for everyone, but I do want to share so that you can take them into consideration. So about research, I think there are two things that are super helpful. And one is trying to be a T-shaped person, meaning that you have some skills that you are sort of unique. You can drill really deep down. And also you have that width or breadth in your understanding of different disciplines, because that's kind of related to the second point as well, be collaborative. I found in my experiences, collaborations with different people in different regions, different backgrounds, super rewarding. And about mentorship, I think that's so important in the process of our career development at different stages, honestly. And I think the first important thing to keep in mind is to take the initiative. Sometimes you need to take the first step to approach to mentors, ask if they could be your mentors. But that also means you need to be prepared when you approach someone and you want them to be your mentor. Basically, ask them to donate their time to mentor you. You want to be prepared. You are ready to discuss the specific questions you want their help with. And the second is just observe and learn. I think it's not necessary for you to develop a very official mentorship relationship to get mentored. That's not necessarily the case. When you see someone as a potential mentor, I think one easy thing you can get started is just observe what they're doing or how they are handling things and learn from that observation. And the third one is climb and reach. So basically, in the process of climbing the career ladder, I think we should also keep in mind that we should look down and try to reach to other people, to help other people. So it's always kind of a two-way street. You get mentored by senior or not even senior or your peers, and you can also serve as a mentor to people who are undergoing things that you probably have gone through. So about service, meaning service to other professional societies or editorial boards and etc. I think those are very valuable experiences that help put your own work into perspective. It really helps to broaden your horizon. And also, I think it helps with professional networking. Sometimes it's professional networking is not necessarily like meeting people with that specific goal. I think it could be built in your experiences of serving on different boards or different groups. And the third one, which I think is very important, is that in those service roles, you help actually create a better working environment because you have a role to play in shaping that. So I think that's also very important. I think when we think about science, we often ask ourselves to be curious. And I think in another way to put it is be open-minded, not only about our research, our work, but also about every other aspect in the process. So that's all of my presentation, and I thank you for your attention. And I'd be happy to take any questions after the second presentation. Thank you.
Thank you very much, Hein. This is really exactly what I think we would like to hear. It was wonderful, your presentation, your tips. And there are questions. There are already questions from the audience, but we'll save them for the end. So now we're going to welcome our second speaker. So if we can add Amir to the stage. Yes, there he is. So Dr. Amir Pahlavan joined the Department of Mechanical Engineering and Material Science at Yale as an assistant professor in July 2021. He earned his PhD in mechanical engineering from MIT and then moved to Princeton University as a postdoc, working in the areas of interfacial flows, transport phenomena, and hydrodynamics. Inspired by geophysical and biological flows, his lab explores a range of problems on active and passive transport phenomena in complex environments. So Amir, the floor is yours.
All right, thank you, Marcel, for the kind introduction. Can you hear me well?
We do, very well.
Oh yeah, perfect. All right, so first let me thank the organizers for this opportunity. It's really an honor to be part of this series. I've watched many of the videos over the past 2 years or so, and I've learned a lot. So I'm really honored to be part of this series. So today I'm going to just give a very short presentation on some of the work we've been doing here in my group at Yale. As Marcel mentioned, I've been here now for the past 2 years, and I've chosen this movie because it represents the why, the beginning of Yale. So I thought because we are relatively new, I'll just give a very quick overview of the type of things we do in my lab here. So broadly, I think one way I can characterize our research as interfacial transport phenomena in complex environments. And there are three thematic themes that are going on in my group. One is related to Faraday transport of colloids in complex environments that I'll be talking about today. The other one is about interfacial instabilities in confinement in colloidal suspensions. And the third one is about the bacterial chemotaxis, collective behavior, and biofilm formation. So just to give you an example of our work on colloidal suspensions and interfacial instabilities, here is one example that shows what happens when you put these and the suspension of nanoparticles in confinement. So you have this confined geometry that is like few hundreds of microns or like in a couple of hundreds of microns thick. You put these nanoparticles in, it's open from one side, it starts evaporating. As it evaporates, it brings in the particles from left to right. You end up getting a jammed solid here on the right. And that jammed solid is under huge stress because of these capillary menisci that are forming at the nanoscale because the particles are nanometric in size. And that leads to formation of cracks. So let me show you what you actually will see when you put the suspension under microscope. So what you see is this stick slip kind of behavior of the crack front that keeps going forward. So of course, we are not the first ones to look in this problem, and a lot of people who work has been done over the years looking at a similar system. So what we are asking here is can we control, suppress, or enhance or modulate the crack patterns? So that's one of the questions we've been looking into. The second theme that my group is working on is related to bacterial dynamics, specifically on biofilms. So what we are asking is that how biofilms form in complex environments, including porous media, and how they evolve in those environments. And there has been beautiful works in the past couple of years coming from different groups. I've just listed a few examples here. There are much more. But this is a multi-scale problem. You start from micron scale dynamics of an individual bacteria. So these are dilute. You don't really have collective behavior here, but then they come together and form these structures that you can call them living elastic media, sort of poroelastic media. Or you can put many of these bacteria together, and then when they realize that lots of them are sort of nearby, then they start showing these flows that are on scales that are much larger than the scale of an individual bacteria. And there has been a lot of research from active matter community, looking into this problem, trying to figure out how these collective behavior emerges. And the aspect that we are looking at more specifically is that how does confinement affect these collective behaviors? So here, what you're seeing is the example of lots of these bacteria when they are put in a droplet, how they behave. All right. So today, as I mentioned, I'm going to focus on our work on phoretic transport in porous media, and our porous media is microfluidic chips. Really, it's not real porous media. And I, by the way, forgot to mention. I think I had the pictures up as I was presenting. The people who are responsible for most of the work that has been done are my group. Why do we look into this problem? What motivates us to look into dynamics of phoretic particles in complex environments? So I would put that theme under this umbrella of gradient sensing in noisy environments. So the main question we are trying to ask there is that how do colloids and bacteria, so I would categorize them as passive and active colloids, respond to chemical gradients in dynamic and heterogeneous environments. So why do we care about that? So I've included two examples here. In the context of ocean, you have these marine snow or these organic matter that are sinking to the bottom of the ocean. There has been beautiful work from multiple groups, including Roman Stocker's group, that have looked into this problem and sort of try to understand how the interaction between bacteria and these sinking, dissolving organic matter affects the global carbon cycle. On the other hand, you might think about the dynamics of these microbial communities in soil, how they interact with plants, with the root system, with fungi networks, and how that affects the global carbon cycle. In both cases, a fundamental question that keeps coming up is that how do bacteria detect gradients, and how do they use that to navigate their environments? Similarly, if we don't go beyond the environmental issues, in the context of biology or our human bodies, more and more work has been done over the past decade or so on sort of trying to come up or engineer coming up with synthetic systems that allow us to deliver drugs or do operations inside our bodies, like with micro and nano robots. And an important question there also has been that, like, how do we how do these robots, when they go to these smaller scales, how are they going to navigate their environments? You are dealing with a very complex environment they need to navigate through. They cannot have a camera at that scale, right? So they cannot really see. So how can they sense their environment? And again, gradient sensing comes up as a mechanism there for a multitude of applications, including the drug delivery to tumor cells, for instance. All right, so let me jump into what I was going to talk about, and I'm going to talk about, and in the context of diffusiophoresis. So what is diffusiophoresis? Diffusiophoresis is the motion of colloids due to chemical gradients. This is not something that has been invented recently. It's been around since 1950s, going back to the Soviet literature by Derjagun and others. And then later on, in around late 80s and early 90s, it was sort of I guess, taken up in the Western literature by Anderson and Prevey and many other people. And the basic idea is that if you have a charged colloid, it doesn't have to be charged, but I'm going to focus on charged colloids. If you have a charged colloid, and if you have a gradient of concentration of, let's say, an electrolyte in the bulk, again, it doesn't have to be electrolyte, but most solutions are ionic, so therefore this is sort of very generalizable. If I have a gradient of a solution, just a salt concentration or solid concentration, that is enough to drive this particle into motion. So the particle, there's no flow, the particle starts moving. So why does it move? It's typically sort of characterized as the sum of two effects. One is called chemiosmosis, and the other one is called electroosmosis. So I won't go into details, but the chemiosmotic effect is due to entropic interactions between ions and the surface of the particle. So you have some interaction going on, and that usually points the flow from higher concentration to lower concentration. And then the electroosmotic contribution comes from the fact that when you have an ionic solution, you have cations and anions. They typically diffuse at different rates, and depending on how they diffuse, they generate these local electric field next to the surface of the particle, which drives another flow. So taken together, you can basically drive these equations, and then what you find out is that velocity with which this particle moves, you can write it as a coefficient that you can take it to be constant. For simplicity, we call it gamma p, diffusophoretic mobility, times gradient of logarithm of the solid concentration. So you have this grad log C term. That is very important because so you would note that it's different from grad C dependent. So it gives you interesting features. So as I said, this effect was known in the colloid chemistry community for quite a while, but it was around, as far as I know, the very first work that was utilized this effect in microfluidics was around 2008, where Lederic Bouquet's group showed this the utility of diffusophoresis in a very simple geometry. So what they had was that they had this microfluidic channel with 3 inlets. The 2 sides are buffer solutions, and then they injected these fluorescent colloids in the middle. So what you see here at the bottom is basically this fluorescent signal that you get from these colloids, and they are plotting that fluorescent signal as you go down down this channel. What you see is that the width of this fluorescent band doesn't really change because particles, the diffusivity of particles is typically very small, which means that the particle number is very high. So then they don't really diffuse much. But then they did this beautiful trick that they just added a tiny bit of salt to these two buffer solutions on the edges, and along the same distance, then this band, they were able to either expand this band by a huge amount, or the other way around. If they added the salt in the middle solution, they were able to focus that band quite a bit. So that was that gave rise to a series of investigations that it just keeps growing in time. That researchers started using diffusophoresis to manipulate colloids, to make them go into dead-end pores, or get them out of dead-end pores, or into grooves or out of grooves, or try to focus the particles in certain environments, certain areas, or even like, going down to smaller scales, trying to manipulate smaller macromolecules, or deliver particles to hidden targets. And more recently, it has also been shown that diffusophoresis might be responsible for cargo transport within cells. So it has, this field has really exploded in the past 10 years or so. But one question that really hasn't been addressed so far is that all these examples that I showed you are in very simple environments and simple geometries, and they typically involve very sharp gradients that are imposed externally. But if we go again back to the complex environments that I motivated my talk with, from human body to subsurface environments, you typically have lots of gradients, but they are in a very complex environment. You have typically flow, you have mixing, you have disorder in your environment. So that's motivated us to ask the question, how would this interplay between flow and mixing and this heterogeneity in the environment influence this diffusophoretic effect? Is it going to even remain strong and play a role in porous media flows? So we are asking them two specific questions. Do these gradients, solid gradients, remain strong enough at the porous scale when you have lots of mixing in porous medium? And even if that's true, when you have these sort of random array of pores, would their effect cancel out, or would they sum up to something that would have a macroscopic effect? So to answer that question, what we did, we started with these 2D microfluidic geometries that we put an array of obstacles inside. And we first filled this microfluidic geometry with colloids that are typically 1 micron in size, and background salt concentration, C0. And then we start injecting a different salt concentration upstream to displace the colloids and the existing salt. Okay, so this is a single phase flow in the sense that you have only an aqueous system and you have colloids, right? And then we look downstream somewhere here and basically try to observe if we see any sign of this salt gradient playing any role in our system. So first of all, as you all know very well, you have this disordered medium, you get this disordered flow field, you have these preferential flow path and these low permeability pockets. So you have this heterogeneity in the velocity field, right? And we also know that when we are thinking about transport and mixing of, let's say, solutes or colloids, especially if you're thinking about colloids at very high particle numbers, especially in 2D, then their transport is going to be limited by streamlines, right? They cannot get beyond these streamlines if you neglect diffusivity. They cannot hop across these streamlines. Whereas now, because of diffusophoresis, if you're thinking about transport of these colloids that are coupled to this solute diffusivity, they can actually experience an extra force that allows them to hop across these streamlines. So let me try to first answer the first question I raised, that let's go and focus on a dead-end pore with it deep into this porous medium, and ask the question whether this diffusophoretic effect is relevant. So here I'm showing you 3 experiments. The control one is a case where the salt concentration we are injecting is the same as the salt concentration that is within the domain. The attractant case is a case where the salt concentration we are injecting is higher, and the repellent case is the vice versa. So what you can clearly see is that these particles start going out in this case, and particles start going in this case. So I've included this diffusion time scale sort of as an estimate for the control case, that if you wanted to wait for these colloids to diffuse on their own, let's say if this pore is 1 millimeter in length, diffusivity of these particles around 10⁻13, you have to wait 4 months for these particles to get out. Whereas using these salt gradients, now we are showing that even though you have a lot of mixing, you can make these particles to get out or into these diluent pores on the order of seconds, right? So it's much, much faster. And the reason is that you are coupling the diffusion of these colloids to diffusion of ions that can diffuse much faster. So we can do a little bit better than that, right? So we can do dual channel imaging both the solute front. So we've added fluorescein in this case. So what you see is that the red color here is representing the salt front and the colloids. Maybe if I can, let me play this again, if I can. Yeah. So what you see is that when the solute front comes in and the red color shows up, you can immediately pick up on the effect that it has on the particles. So that also shows sort of as a further evidence that the effect we are seeing is due to solute front. So now if we ask a question on a macroscopic scale, so we take a bigger picture and ask how, say, in this case, these are fluorescent colloids. You can either count them or you can just take a look at the light intensity, and you see how that light intensity evolves in time. What we see is that in the control case and repulsive case, they sort of behave almost similarly. So why the repulsive case is behaving similar to control case is a question that I can talk about. But the attractive case shows that you can remove these particles much, much faster than these two other cases. So on the order of, let's say, in this case, 200 seconds as opposed to, so this one doesn't get to this level within our experimental time scale. So if I look at this as an effective 1D system with an effective 1D equation describing the colloid concentration, what I find out is that I can define a macroscopic dispersion coefficient, and that macroscopic dispersion coefficient is changing by a factor of 10 or so, showing how diffusophoresis is affecting transport of colloids. So this is, I think, very interesting because so far the classical description of transport of colloids in porous environments has not accounted really for these solute gradients. And what we are showing is that these macroscopic dispersion coefficients can vary quite a bit due to these solute gradients. So I think this is an exciting field, and we are probing this more. So this is all the story I wanted to tell you about. So I should thank my group and the funding agencies for funding our work. As the second part, I should also mention my career path. My career path, unfortunately, I don't have as many details as the beautiful talk that Han gave. I'd be happy to talk more about it. But I did my undergrad in University of Tehran. I grew up in Tehran, and I had a few professors who gave me opportunities to work in fluid mechanics. I got interested in fluid mechanics. Later on, moved to University of Illinois, Urbana-Champaign. Initially, I wanted to do turbulence research, and then I didn't get into that field by accident. I ended up going to low Reynolds number, which for fluid mechanicians, they know that's sort of exact opposite of turbulent flows, and I loved it. So I continued on it. So then for my PhD, I ended up going to MIT and worked with so I'm not naming names here because I'm sure I'll forget some of the names. So because of that, so with people and I learned a lot, and I worked on multi-phase flows. Started from theoretical side modeling, then gradually found my way into the lab, and annoyed people enough in the lab that let me play, and then later moved to Princeton, doing my postdoc there. I continued in the same realm, so tried different things, and started this colloid work and bacterial work. Some of it started there, and I've been at Yale since 2021. So if I really don't have much advice. I'm still, I consider myself early career, so I'm not in a place to give advice really, I think. But two things came to my mind thinking about this. One is that definitely it's about mentors, collaborators, and community. really takes a village, and I've been lucky to, throughout my career, to have the opportunity to interact with really amazing people and learn from them. And the second thing that this is an advice I try to give myself from time to time is from Einstein, and apparently that's in a letter he wrote to his son. Um, that life is like riding a bicycle. To keep your balance, you must keep going and keep moving. And I've tried to apply that advice to my own life. So with that, I'm done. Happy to talk more.
Super, Amir, thanks a lot. That's wonderful. I'm going to, we have like lots of questions for the audience, and we're going to have a little bit of a discussion further ahead. So I'm going to call my friend Muhammad to help a little bit with the question rounds first.
Yeah, excellent talk, Hong and Amir. I personally really enjoyed it. But I will start with I have a couple of questions of my own, but I will start with the questions from the audience. The first one is from Gigi, and it's directed to probably Hong's presentation. Have you considered using a pore network model for numerical simulation as hybrid regime between the pore scale and continuum scale? What are the advantages and disadvantages of both, according to you?
Thanks for the question. So personally, I haven't done any work using pore network model, but I've known colleagues who've done that. So there are definitely advantages and disadvantages to the hybrid approach that I've shown and the pore network model. I think it's kind of a balance of the computational cost and also the level of accuracy that you kind of value more in your specific problem. So yeah, it's always a trade-off.
Excellent. The next one is from Koirin. Hi, is there a length scale associated with the localized low supersaturation precipitation?
So I guess that's my question as well.
Yes,
so associated with localized low supersaturation precipitation. So there's two length scales associated with that. One length scale is kind of from the inlet to where you observe the precipitation. That's on the order of hundreds of micrometer for the given velocity in the experiment. And the second length scale is the thickness of the precipitating layer that's replacing the substrate. So in that case, it's on the order of 25 micrometers, as also reproduced in our model. Yeah.
Thank you. A comment from Right the Himalaya. Excellent insight, Han. Thank you for your talk.
Well,
the next one is by. The next one is from Negar, and she's asking you, could you please elaborate on the key parameters that play a significant role in determining critical effective diffusivity?
So I wouldn't say that I understand the systems well enough to name all the key parameters, but based on our observations, it actually goes back to nucleation and crystal growth again, because in order to determine the critical effective diffusivity, it is important to account for the microporosity and the diffusion process through the precipitation itself. And that texture, as I mentioned in my presentation a little bit, is controlled by the nucleation process. But again, if we step back and thinking about this system, the critical effective diffusivity is going to be controlled by the pore structure, the initial pore structures, because that's controlling where the precipitation is going to occur, and also the pore throats that's going to actually clog the system.
Excellent. And as a follow-up, she continues, have you observed any notable impact on the precipitation rates when the diffusivity is updated?
Okay, so that's about the continuum model. Like if we implement the new diffusivity, do we see any impacts on precipitation rate?
I would say yes.
Yes, so if I understand that question correctly, that the answer is yes. So actually, as the diffusivity decreases sharply, so does the precipitation rate, because it's a diffusion controlled system. Once we turn off the mixing, the precipitation is slowed down.
Yeah, okay. And now a question for Amir from Gigi. Is there a way to tune the precipitation as a function of porosity? Is there a phenomena, or is there a phenomena of clogging, unclogging in general when using mesoid particles in aqueous phase?
So I didn't really talk about precipitation, so maybe that part is directed at Hong. I'm not sure, but generally about clogging and unclogging, that's a great question. So we think that these solid gradients do affect these processes as well, and we've started working on that too. And I don't have an answer for that yet, but that's a great question.
Okay. One question for Han. Thank you for sharing your research work and insight. Could you compare your experience in national lab and in the university?
Yeah, sure. So I think there are a couple of major differences. One is in terms of teaching. So in national laboratories, we get the opportunity to mentor students, especially undergrad interns and etc. But there's a rare opportunity to kind of mentor a graduate student throughout the entire PhD program. So that's kind of very different. And also in terms of teaching, you probably get asked to give guest lectures in classes, but developing a course and teaching a course from the beginning to the end, that's sort of unique to the university. And in terms of research, I think the difference would be the access to probably facilities, because national laboratories are known for their very fancy user facilities. They are, of course, open to all researchers across different universities and etc. But being physically in the lab always get the advantage, because sometimes when an applicant kind of bails out or was not able to make it, that time can be filled by local researchers. So that's another difference. And also, I think a difference which probably is decreasing as we move on, is that in the national laboratory, the research is definitely more collaborative, because the one of the initial goals of establishing national laboratory is to have team science. Basically, they encourage people to work on problems that can not be easily solved by a single or a small group of researchers. So that's kind of the default mode of working in national laboratories. But I understand that nowadays in universities, there are so many cross disciplinary centers, and also big projects being awarded to fund multiple universities. There's that type of opportunities as well.
Okay, excellent. It was a question by Song Lee, one of our co-organizers, part of the Porous Media team. Thank you, Song and Hai. And okay, I guess we have no more questions from the audience. Now I can ask my question from Amir, or Marcel, you can go on first, and I can continue.
Okay, super. So one quick question for Amir as well. So you showed, like, in the beginning this kind of like schematics and the experiment result, when you had like the solution with salt coming from the sides, and then like the one without from the center, right? And then it tended to spread out a little bit more, and then there was like another one where it did the opposite, and it tended to focus a little bit more, right? And that's kind of like made you think, okay, so these processes are kind of like one is the opposite to the other. But then later on, you showed like this plot where like the behavior of the repulsive was very similar to the one that was without any kind of like a solute, and the attractive one was like super, super fast. So where does the symmetry get broken?
That's a great question, Marcel. So it's hard to answer that question, but let me put it this way. The easiest way to sort of, I think, for me at least, to think about that is that in the first context that you are correctly referring to, in that case, you have flow from up to bottom, right? And then the diffusive effect was happening in perpendicular direction. So in that direction, you don't really have a flow, right? So then diffusive forces is doing its job, right? And striving things either this way or the other way around. Whereas in the example that I showed you, we are looking at these pores that are all over the place, and you have a background flow that is going in that direction, like from left to right, and pores can be in many different directions. So there you have to think about the competition between diffusive forces or diffusive velocity and background fluid velocity. And that's really at the heart of the fact that you end up getting different effects. So to make it simpler, or I'm oversimplifying this, but if you just take a dead end pore, right? If I impose a solute concentration gradient across it, before that solute comes in, you don't have any motion, so there is no convective flow. So it's just a solute that is driving the diffusive motion. So nothing is sort of there's no convective flow to block it off. Whereas now, if I'm thinking about the repulsive case, when I want to push a particle into that dead end pore, the particle is being advected in the back by a flow. So that diffusive forces has to overcome that background flow and push it into that dead end pore. So that's sort of, what leads to this. But it's more complicated than this simple answer, I think.
Another question for Amir. Do you see any possibility to use the diffusivity transport to, for example, heal cap rocks or heal cap rocks, or I don't know, the sealing layers in the storage sites for the nuclear waste, for the CO2 storage sites, or things like that? Can we design any kind of fluid or any specified particle to do a certain job in a localized location?
So that's another great question. I should admit first that I know next to nothing about the two problems you mentioned, but I sort of first of all know that there is now activity in the Europe actually of people thinking about delivering particles to certain sort of locations that are hidden in your porous medium. But in principle, I think the answer is yes, right? So if you know, if you can design, you can have a source of reaction, right? So like, and that source of reaction is driving some solute gradient, and then you can inject particles that are sort of like to move up that gradient, and by that, as a result of that, like you can address some of the things you mentioned. But then you can also pose this question, I think, more generally, and ask the question like, sort of for whatever kind of solute, what kind of particle should I design if I wanted to design it? And I think, but that probably would be too expensive in the context of soft surface flows. But in the context of biological applications, I think that might make sense. For instance, like in the context of drug delivery to tumor cells, right? So you can sort of do that, and people are doing that. And maybe you can use similar mechanisms to deliver drugs to these tumor cells that are hard to reach.
Excellent. Do we have any more questions from the studio? Federico, Mahdi?
I might have a question for Deng. If I remember, in the second part of, if I remember correctly, from the second part of your talk, one of the main message was the fact that with respect to the single phase, in the two phase, the reaction rate was impacted much more. If I understand correctly, this is due to the presence of foam. So the mixing in a microscopic sense of the air and the liquid in some sense, or the two phases in general. So what about if I don't have foam? So if, for example, the second phase invades the first in continuous path, for example, if you studied, of course, do you expect a particular impact as well on the reaction rate?
Okay, so I should first clarify that in the example, I should have shown the two-phase flow. In the second example that I mentioned, it's also not a foam system. It's just gas bubble flowing through the system. So I would, regarding to your question, as if I have a phase invading into another phase in the more sort of dispersed form, how would that impact reaction? I think in that case, first of all, probably there's no effect through surface area because it's going to be mostly in bulk, but those are educated guesses. And the second, in terms of the transport, so I think it could be mixed because in one way, in our specific geometry and systems, we observed transport limitation. But I could also imagine that it could actually improve mixing because at the interface between the two phases, sometimes you see higher velocities. So that could actually, in some, depending on the reactions and also the systems, that could indeed actually improve the reaction. So I think it will depend on the specific reactions and the system, but those are, I think, different possibilities for the system you just described.
Thank you very much. Very interesting.
Well, thank you.
I've got a question from Hang, if I may.
Sure.
At some point of your presentation, you were mentioning to compare with experiments. You just took one stream through the experiment and like did the simulation. Wouldn't that oversimplify the fact? Wouldn't that tortuosity of the porous media and like the correlation between the next stream lines, wouldn't that affect the results?
So that's an excellent question. I did not explain details. So first of all, we did account for tortuosity in our setup. So basically, the porous media column is about 1 centimeter, whereas the streamline we simulated is 5 centimeter, and that factor of 5 is determined by the measurement of the effective diffusivity of the column. So that's kind of reduced compared to the molecular diffusion by a factor of 5. That's kind of indicating, sort of coarsely indicating that the tortuosity is on that order or by that factor. So that's how we sort of accounted for the tortuosity. But you're right, we did simplify a lot of things by assuming just to follow a streamline. I would imagine that when you have different porous roads and geometries along the flow, that could affect local velocity profiles quite a bit. So we were able to kind of do the comparison, sort of coarse comparison along the flow direction, but not our match is not 100% quantitatively. So yeah.
Okay, thank you very much.
Yeah.
Excellent. Just to wrap up the session, I have one last question to both of our speakers, and it's sometimes in academia it can be lonely. It's hard. It can be hard to find your way and to decide to stay in academia or not, and what is the best way forward, and how to get the support you need, and how to build the network that you want, and all these questions that can accumulate, and it's hard to find answer for them. Do you have any insight or any comment on such kind of questions that PhDs, postdocs, and many of us in academia can feel and experience?
I can take the first one stab at answering that question. So I think there are different supporting networks. So first of all, you have your advisor, and sometimes I understand that, especially when you are going through the PhD program, it's not always that you and your advisor are like the honeymoon period. So sometimes I think it's also helpful to reach out to your committee. So I think there's a reason to have a committee instead of just your PhD advisor. I think in a lot of the cases, the committee members are very willing to help as well. So some issues or problems you're reluctant to take to your PhD advisor, you can probably approach the committee members trying to get some input. And the other important network is, I think, the cohort of the graduate students. because you are going through the same process at the same time. And I think the support that you can get from them is probably very specific to you, and probably more valuable compared to others. And I would also say that sometimes when you're feeling lonely, it's I think it's also important to realize that it's not necessarily a very scary thing. I think sometimes that could be it could be the starting point of a negative spiral or feedback, but that's not necessarily the case. I think sometimes, at least to me, it's probably helpful to actually take some time to be alone and to go through the processes of trying to think about what motivated you to get started in the first place, or how you want to kind of develop your career for future. Is there alternative, or what's the worst that could happen? Sometimes if you think about a worst scenario and you are like, okay, that's not so bad and I can live with it, it probably takes some pressure off. So I think it's fine to encounter that type of period and try to process that. But of course, if that's like working on yourself is not working, as I mentioned, it's helpful to approach to your committee members, your fellow graduate students, and of course, family. They're always there for you, for sure. Yeah. Hopefully that helps.
What about you, Amir? Do you have anything to add?
Yeah, I was gonna say I'm not sure if I have much to add to Hang's answer. Yeah, I don't think there is any recipe that would fit everyone. Everyone's situation is different. And academia, as we all know, has many tortuosities, right? No pun intended. Like, the path for everyone is very tortuous. I think, it depends on what stage in your life you are, the decisions you're gonna make. But having a community, such as this one, which is a very friendly community, and I found it very welcoming, I think is definitely very helpful, because then you can talk to people and sort of ask about their experiences, and then you won't feel alone as much, because in especially like PhDs and postdocs, sometimes we are frustrated because we are not making enough progress. There is always comparisons that people make with others, and that could add to the difficulty. But then when you realize you're not alone in this, right? Sort of you have a lot of people that are in the same boat, that helps a bit. And I would only say that as long as you think that you're in a healthy environment, right? So it's, things can be hard, but then if it can be unhealthy, if it's not unhealthy, that's fine, right? So then, maybe you can overcome that. But if it's an unhealthy environment, then you need to get help and sort of try to find a way out, sort of. Yeah, that would be my 2 cents.
Thank you very much. Can I hand it to Marcel for closing the session?
Yes, thanks a lot, Mohammed. This was a lot more than the 2 cents from both of you. I think we benefited a lot, like not only the audience, but us in the studio too. I for sure did. So thanks a lot to both of you for taking the time to participate in this session. It's the longest Tea Time Talk session that we have, I think. We're like much beyond the 1 hour limit, but there's a reason for that. So I want to close by finishing. First, I want to add to show a little graph that we had made. So Chiara from our team, she made this little word map here with all the universities that the Tea Time Talk has had participants from up to the previous session. So this is we have 2 more now on the 50th session. And you see, like it's a pretty spread. I mean, there's a lot of blue in the graph. But for sure, there's a lot more that we need to cover. We would like to see on the second 50 groups of sessions now starting to spread a little bit to the gray area of this graph. Also, like our balance in terms of genders is now 70% male and 30% female, which is perhaps a little bit representative of the porous media community as a whole, but we want to get better at that as well. And you see, the thing that we have a big bias is geographically, there's a lot more of European talks than people from outside of Europe. And that is because many of us are in Europe, and the community, the people that we know from, that we can invite are also either in Europe or mostly in the US. So we need some help here from the whole community to help us address the problems here, to get it more diverse and more inclusive, both in the matter of the genders, but also with location, with where people come from. So please do help by suggesting speakers. You can send that directly to us. You can contact the speakers first beforehand if you're comfortable with that. And that will help us a lot, so that we can get even more diverse and inclusive on the second part of, I don't know if it's the second or third or fourth part of the Porous Media Live. We don't know how long it's going to go. Like now, it's completely open of the Tea Time Talks Live. But yes, so thanks a lot once again. And I just want to finish again, showing the whole team here, the present and the past members. And if you want to reach us, you can contact us anytime by Gmail at porousmediattt@gmail.com. And either to say anything that you think we should improve, or like suggest speakers, we're open for conversation there anytime. So thanks a lot to both of our speakers and to the whole team from today. We'll see you very soon. Bye-bye.
Thank you. Bye.
Bye.